So it's with great pleasure that I am here to introduce Emily Balskus. She's a professor of chemistry and chemical biology in the Department of Chemistry and Chemical Biology at Harvard, where she has started a long time ago, but not too long actually, as a PhD student, and then joined in as a postdoctoral fellow, and then became assistant professor, associate professor, and now professor in a very, very short period of time. So an amazing, amazing career. I have here a long list of awards. So you see a lot of pages. I'm just going to go through some of the most recent. She was awarded the Tetrahedron Young Investigator Award in 2019. She's a laureate in chemistry of the Blavatnik Award for Young Scientists. She had got a number of awards from Eli Lilly, from Pfizer, and really the list just keeps on going and going. So I'm not gonna take time from her talk. She's focusing on how really understanding the chemistry of the microbiome and understanding how that affects health and disease in humans. And we're gonna hear about that, but it's for me a particular nice moment because she contacted, or actually we got in contact through our common friend, Shana Sturla, in 2015. So August 21st of 2015 is when we started connecting. And since then, we have been working on a number of grants and a number of projects that have been really very successful and have brought our DNA damage work into the microbiome world and kind of in a unknown territory from where I started. So thank you for that. And thank you for being here today, Emily. I'll let you start your talk. Thanks.
Okay. Well, thanks so much for the kind introduction, Sylvia. And thank you all for the invitation to be here. I'm very excited to share some of my lab's work this morning. I'll also be around for a few days. I'm staying until Friday morning. And so if anybody wants to follow up with questions or conversations afterward, please let me know. So I actually was trained as a synthetic organic chemist as a graduate student. And what originally drew me to microbes were really their amazing chemical capabilities. This continues to be a source of fascination for my lab, simply wanting to discover fundamentally new enzymatic chemistry and metabolites from these organisms. But since starting my independent program, we've also become very captivated by the biological roles that microbial metabolism plays in complex microbial communities like our own human microbiome. So microbial metabolism expands the chemistry of the human body and can be correlated very strongly with health and disease. But there are currently major gaps in our understanding of the metabolic activities of the microbiome that limit our understanding of this community's impact on host biology and our ability to intervene therapeutically in the many diseases linked to these organisms. So it turns out that most metabolism linked to the microbiome has not yet been connected back to specific organisms, genes, and enzymes. Also, as you'll see, a huge fraction of the genes associated with these organisms and the metabolites that they are producing have just not yet been characterized. And we're only really just beginning to understand the molecular mechanisms by which microbial metabolism influences the hosts and might be affecting the rest of the microbiome. And so the goal of research in my lab is really to try to use chemical knowledge and tools to address these gaps. We seek to discover new enzymes and metabolic activities from members of the human microbiome, as you'll see, with an emphasis on gut microbes. And we're also seeking to develop tools and approaches that we can use to try to understand how exactly microbial metabolism is influencing host biology. And so I thought for today I would share two stories that have different focuses, I think illustrate hopefully the scope of the types of problems we're interested in, the breadth of different approaches that we use. We'll hopefully have some broader connections to biological problems and questions that interest you. So this first story I'll share really focuses on the question of how do we discover new enzymes that are associated with the microbiome. And this is a huge problem, as I sort of alluded to in the introduction. If we just look at the scope of genes associated with the healthy human gut microbiome, here I'm sharing data that was originally obtained from the Human Microbiome Project. These are gut microbiome sequencing data sets from 150 healthy individuals. And what my colleague at Harvard, Curtis Huttenhauer, is doing in this analysis is just categorizing the genes associated with each individual according to how confidently they can be annotated or assigned a function. You can see that really only a small fraction of the genes in any individual can be given even a tentative annotation. So there's a huge need to connect this genetic potential with actual functional capabilities. So I mentioned that my group is trying to use chemical knowledge to do this. So what exactly do I mean by that? Well, in many of our projects, what we're trying to do is to kind of take a metabolic activity of interest that we know is associated with the microbiome and is not yet characterized at the genetic or molecular level, and link that activity back to genes and enzymes. In other cases, we may be starting with genes of unknown function that we suspect encode enzymes and are attempting to link those genes and the proteins they encode back to actual functions and metabolism. You'll actually see examples of both of these things in the talk today. But what I think my group brings to this problem that's special is our understanding of how enzymes work at a molecular level and our understanding of the chemical logic of metabolism. So you'll see that in much of our work, we combine bioinformatics, mining of genomes, prediction of protein structure, tools like that with chemical intuition and chemical knowledge to try to generate hypotheses that guide a lot of this discovery-based work. And so we've successfully applied these types of approaches to link various types of microbial metabolism to genes and enzymes. So I thought I'd share kind of just a taste of the scope of projects we've worked on. We have studied how the microbiome metabolizes both host and dietary nutrients. So one recent example from the lab is kind of uncovering the first gut bacterial enzyme involved in cholesterol metabolism. This enzyme and its presence in human gut microbiome sequencing data sets is correlated with lower total cholesterol levels in human cohorts, which potentially suggests that this pathway for consuming cholesterol in the microbiome may be having an impact on host cholesterol levels. We've done a lot of work investigating how the gut microbiome metabolizes other types of ingested small molecules, specifically small molecule drugs. One example of this is our work on levodopa metabolism. This is, of course, an important medication for treating Parkinson's disease. And we've discovered some of the enzymes and organisms involved in this process, and also revealed that there's a lot of variability in this metabolic activity between different people, and this could perhaps underlie some of the variability in responses of Parkinson's patients to this therapy. And then finally, in work that we've had a chance to collaborate with Sylvia on, we've been studying how gut bacteria make their own complex metabolites, their own bioactive natural products that have various links to host health and disease. In this case, this molecule colibactin is connected to colorectal cancer development. And so again, I'd like to share a story today about enzyme discovery. And this story really has some links to our work on levodopa metabolism, as you'll see, but really began with an interest in understanding a chemically challenging transformation. So we got into this project really through the lens of chemists wanting to understand unusual reactivity. And so this was work that was started in the lab by a former graduate student, Vayu Mene Rekdal, was continued by a former postdoc at Chip-Lay, and is currently being led by a graduate student, Min-Woo Bae. And the chemical transformation that originally caught Vayu's attention was this metabolism of catechol containing molecules. So a catechol is a 1,2-dihydroxylated aromatic ring. And it was well known for many years that the gut microbiome as a whole could metabolize these types of structures through the removal of a hydroxyl group. So this happens completely selectively, removal of just one of these two hydroxyl groups. And to a synthetic chemist, this reaction is quite challenging. There's actually no direct equivalent to this transformation in synthetic chemistry. In addition to just being neat microbial chemistry, we were also struck by the potential biological relevance of this transformation. So you'll recognize that type of catechol functional group as being an important part of many molecules, perhaps most notably dietary polyphenols, which will come up again in a few more slides. But this catechol group is also found in host-derived molecules like neurotransmitters, and can be also generated through metabolism of in the metabolism of small molecule drugs like this immunomodulatory agent. And this type of metabolism may seem very small from a chemical perspective. We're really just making a small replacement in these structures, but this type of chemistry actually has big consequences for the biological activity of these molecules. It changes the redox behavior of these catechol scaffolds. It changes their bioavailability and can dramatically change their bioactivity. So as I alluded to earlier, we first kind of stumbled upon this transformation when we were studying levodopa metabolism. And so the first step in L-dopa metabolism is decarboxylation of the drug to give dopamine, which is similar to how the human host metabolizes this compound. But it turns out that the microbiome further processes dopamine through this type of catechol dehydroxylation reaction. And when we began, again, like all catechol dehydroxylation reactions, the specific organism and enzyme were totally unknown. We started this work by trying to screen all of the gut bacterial isolates we had in our lab to see if we had any that could dehydroxylate dopamine. This was completely unsuccessful. And so, the challenge Vayu faced in beginning this work was, even harder than in some of our other projects. We, not only had to find an enzyme, but we had to, find an organism that could perform this metabolism. And so he took a pretty clever approach in starting this project. He had the hypothesis, which I'll sort of discuss in a little more detail in a few moments. He had the hypothesis that perhaps this transformation, which is reductive in nature, might be helping certain gut bacteria grow. And so he decided to try to obtain a dopamine metabolizing gut bacterium by cultivating it from a human fecal sample. So he started with a sample that we knew had dopamine dehydroxylating activity, passaged that complex sample in a minimal medium that had dopamine as a sole electron acceptor, and then ultimately obtained single colonies and screened them for activity, and was able to grow a dopamine dehydroxylating strain, which turned out to be a member of the actinobacterial phylum called Agarthella lenta. So Agarthella lenta, or E. lenta for short, is a gram-positive obligate anaerobe. Members of this genre have been actually quite prominent in microbiome studies in recent years, because it turns out that Agarthella strains metabolize lots of different dietary compounds and drugs. So with a strain identified, we could now move forward with trying to discover the actual enzymes involved in dopamine dehydroxylation. So to do this, Vayu faced the challenge of there not being great chemical precedent for this type of metabolism. So it was hard to mine the genome and rationally identify potential enzymes. But what he had noticed in some of his work cultivating this strain was that actually, like many microbial metabolic activities, dopamine dehydroxylation was inducible in the presence of substrate. So the organisms turned on this pathway and expressed the enzymes in response to growth in the presence of substrate. And so this led us to use RNA sequencing as a tool to discover these enzymes. We simply grew the E. lenta in the presence and absence of dopamine and compared the gene expression profiles. And what you see here, when you look at the comparative analysis of the RNA-seq data, is that there's a very specific transcriptional response of this strain to dopamine, with one genetic locus very highly upregulated. When we look at the contents of this genetic locus, we were very excited to see that it was predicted to encode an enzyme, and specifically a metalloenzyme that was predicted to contain this beautiful molybdenum cofactor. So enzymes of this general class were well studied in the context of other types of microbial metabolism, with the most well-studied member of this family of molybdenum enzymes being DMSO reductase. And so DMSO reductase reduces dimethyl sulfoxide, DMSO, to dimethyl sulfide. So again, a reductive transformation, and this allows organisms to use DMSO as a terminal electron acceptor. And looking more broadly at the superfamily, you see that many members of the superfamily are catalyzing reactions that involve transfer of oxygen atoms. So suddenly this potential enzyme makes a lot of sense as a candidate for dopamine dehydroxylation. So of course, what I showed you was a pretty large genomic region. What is it encoding? So we would predict, based on the annotations of the genes in this locus, that one gene encodes the catalytic subunit of the enzyme that contains the molybdenum cofactor. But there are various other subunits that are also likely associated with this enzyme that are involved in electron transfer or anchoring within the membrane. There's also genes encoding chaperones that may be involved in protein folding and insertion of the metallo-cofactor. Okay, so we needed, obviously, to confirm that this actually was the enzyme responsible for dopamine dehydroxylation. This turned out to be quite complicated. So these types of metalloenzymes were known to be very difficult to heterologously express in other organisms. And that turned out to be the case for us. We tried over 30 different constructs for expression in E. coli. We tried expressing in several other closely related heterologous hosts and had no success. And so what Vayu ended up doing was using activity-guided purification to purify the dopamine metabolizing enzyme out of E. lenta, and so he could identify it and confirm its identity. So this was challenging. This enzyme is air sensitive, so all of the purification steps had to be done under anaerobic conditions. This is a gel from a very early attempt. You can kind of see how little of the enzyme we're able to get out of these preparations. But we were able to purify enough to do gel band sequencing and confirm that this dehydroxylating enzyme is indeed the dopamine dehydroxylase we had found through RNA sequencing, and assays of these enzyme preparations clearly show activity towards dopamine. Now, how is the enzyme actually working? Again, I mentioned this is a chemically challenging transformation. We can look at what's known about DMSO reductase to propose a potential mechanism. So I should highlight that we have very little experimental evidence for this mechanism due to the challenges we've had with obtaining purified protein. But I think this is a starting place to think about future mechanistic experiments. And so I'll just highlight some of the interesting features of this proposed mechanism. We'd start with this molybdenum 6-oxo intermediate. This would need to be reduced to a molybdenum 4 intermediate. We propose that this key intermediate, which is likely ligated by an amino acid from the enzyme, is the intermediate that substrate binds to. We propose that the substrate binds through coordination of the para-hydroxyl group of the catechol. This is the oxygen atom that will end up being removed. We can then invoke tautomerization, so essentially protonation at that carbon atom of the aromatic ring. This disrupts aromaticity. We can then propose an oxygen atom transfer step that would re-aromatize the aromatic ring, break the carbon-oxygen bond, and transfer the oxygen atom to molybdenum. So this could happen through 2-electron chemistry. There's also a potential mechanism that would involve 1-electron transfer steps. This again is providing us with a starting place to pursue some mechanistic experiments. And I'll just highlight what we have been able to do with the small amount of enzyme we have, is to perform assays in buffer containing D2O, and we see that, as one might expect from this mechanism, very specific deuterium incorporation on the carbon atom where dehydroxylation has occurred. So excited to continue to investigate how this enzyme is accomplishing this challenging reaction in the future. But in addition to just kind of confirming the activity of this enzyme, the other feature of its chemistry that really intrigued us was its potential selectivity. So I kind of opened up this section of the talk by highlighting the breadth of different catechols that the microbiome as a whole can metabolize. And so we wondered if perhaps this type of enzyme was just very promiscuous and could metabolize a number of different catechol containing substrates. So we, with our purified enzyme, exposed it, assayed it with a number of different molecules that were known to be dehydroxylated by the gut microbiome as a whole. And what we found was really intriguing, that besides dopamine, the only other substrate this enzyme would accept was norepinephrine, which is actually the most structurally similar to dopamine. So this enzyme, at least, is very selective, raising the question of how these other catechol substrates are metabolized by gut bacteria. I highlight here just some of the catechols that are found in dietary polyphenols. These substrates are of high interest because consumption of foods containing these different types of molecules is strongly correlated with altered risk for various types of diseases. Understanding how these types of molecules are metabolized might allow us to better predict how different individuals will respond to diet and maybe understand some of the mechanisms underlying these health associations. But this observation that at least the dopamine dehydroxylase was very specific made us wonder, could there actually be dedicated enzymes for each of these different substrates? And some initial analysis of genomes seemed to support that hypothesis. And so here I'm showing you the genomes of a number of different organisms that are closely related to Agarthala lenta, so either other strains of Eggerichella or closely related gut actinobacteria. And I'm just tabulating the number of predicted catechol dehydroxylating enzymes that are found in each of these genomes. And you can see that these organisms don't just have dopamine dehydroxylase, they have usually around a dozen different predicted catechol metabolizing enzymes. Some of these organisms, the Gordonibacter strains, actually have upwards of 20 to 30 different predicted catechol metabolizing enzymes, which is a pretty remarkable expansion. So you can see here, if you look at the genome maps of a Gordonibacter strain and an Eggerichella strain, how much of the genome they're devoting to this type of metabolism and these really interesting genomic regions where there's great expansion of this specific type of enzyme. So this suggested to us that probably these enzymes are important for the physiology of these organisms. And I'll just share some experiments we've done to test that idea with the dopamine metabolizing enzyme in Elenta. Again, the original hypothesis that fueled Vayu's work was that maybe gut bacteria were using these compounds as terminal electron acceptors. And indeed, we see that if we compete a Eggerichella strain that can metabolize dopamine with one that is missing that enzymatic activity just naturally, that there is a growth advantage to the dopamine metabolizing strain that depends on the presence of dopamine. So this is perhaps consistent with dopamine metabolism providing an energy benefit to the metabolizing organism. We've also investigated this in more complex settings, taking a gut microbiome sample, fecal sample from an individual that we know can metabolize dopamine, and then looking to see how the abundance of Elenta specifically changes in incubations with those samples. We see that generally in different microbiomes, ex vivo experiments in the presence of dopamine show a increase in the abundance of Elenta as assessed by QRT-PCR. So again, with the early stage in understanding the physiological impact of this metabolism, but we think that different catechols like dopamine may be supporting the growth of organisms. And so having an array of different catechol metabolizing enzymes might be giving these organisms a competitive advantage in the microbiome. Now, with so many uncharacterized enzymes, how do we start linking them to the actual reactions that they catalyze? Well, as an initial attempt to do this, we sort of returned to the idea of RNA-seq, and we examined which potential catechol dehydroxylases were expressed in response to different substrates. So here's 3 different catechols that were metabolized by various gut actinobacteria we had in the lab in our strain collection. Again, we performed RNA sequencing with and without the substrates of interest. And in each case, in the metabolizing organism, we saw high upregulation of just one predicted molybdenum-dependent metalloenzyme. If we then go back to try to see whether the presence of these enzymes encoded in an organism's genome really correlates with metabolism by that organism, we see very good correlation. So this is the phylogeny of the Eggerichella and Gordonibacter strains we have in the lab, their ability to metabolize these different substrates and their carriage of these different genes. So while we haven't biochemically characterized all of these enzymes, I think we have good data that suggests we've assigned their functions correctly. So you might be wondering, what accounts for the very narrow and beautiful substrate selectivity of these different types of enzymes. And so we're in the very early stages of understanding this, but we've been able to gain some potential insights based on protein structure prediction. So like many labs that study proteins, we have become very excited by the availability of protein structure prediction tools like AlphaFold. What we found in predicting and comparing the structures of various catechol dehydroxylases are that there are certain structural features of these proteins that seem to correlate quite strongly with substrate selectivity. And specifically a region of the protein structure known as the substrate funnel. So in these enzymes, the molybdenum cofactor is kind of buried in the middle of the protein, and there is a substrate, there's a tunnel in the top of the enzyme that gates entry into the actual active site. So we've found that members of this enzyme family that process small substrates, like this molecule DOPAC, tend to have very narrow substrate funnels, whereas the enzymes that process larger catechol-containing substrates, like this intermediate in catechin metabolism, have much larger, wider substrate funnels. So this maybe is beginning to help us rationalize the specificities of the enzymes we've already discovered. But as you'll see in a moment, this type of information can also really be helpful in allowing us to predict functions of enzymes with unknown substrate specificity. And so this has really been the question that we've been focused on recently, is, how do we actually understand the scope of catechol dehydroxylation within the gut microbiome, and how do we prioritize new enzymes for discovery? So Minwoo in my lab has been very interested in applying bioinformatics to address these questions. So one of the first things he was excited to do was to take all of the different dehydroxylases that we had linked to reactions and try to assess the scope of enzymes, related enzymes that were present in the microbiome. So he searched various databases, including databases of metagenome assembled genomes. These are organisms that have not yet been cultivated, but are presence we can infer in the microbiome through metagenomic sequencing. He found a total of almost 3,000 catechol dehydroxylase sequences in the human gut. And then he performed an analysis called protein sequence similarity network analysis, using information about the characterized enzymes to try to cluster these sequences into isofunctional groups, so groups of proteins that are likely to perform the same function. And so this is that protein sequence similarity network. There's well over 100 different groups of catechol dehydroxylases. I've colored the groups of protein sequences that we can confidently link to characterized activities in different colors, but of course you'll note how many sequences are colored in gray. Those are all sequences we can't readily assign a function to. So there's a huge wealth of uncharacterized diversity within these sequences. This presents an exciting but daunting challenge. What do we study next? How do we prioritize these uncharacterized enzymes? And so here, Min developed a pretty, I think, intriguing approach that I think might be something that we could readily extend to other types of enzymes within the microbiome. So again, kind of taking this knowledge that many organisms will upregulate enzymes in culture, specifically in the presence of substrates, we wondered whether something similar to what we observe in the lab is likely going on in actual human microbiome. So particularly if we think about dietary inputs, foods that we consume that contain these types of molecules, we would perhaps expect that there would be a specific transcriptional response also within complex human gut microbiomes to individual catechol containing substrates. And so Min started to wonder whether we could prioritize new enzymes to characterize based on metatranscriptomic data sets from human cohorts. And luckily, we were able to team up with a physician scientist who had been working on a very promising cohort. So this cohort, the Men's Lifestyle Validation Study, contained high quality metagenomic and metatranscriptomic data sets, and was also very useful in having high quality information about subjects' diets and lifestyle. With Raj, we were able to start analyzing these data sets. And we're very excited to actually find the catechol dehydroxylases that we had been studying in these subjects. So these enzymes are present in human gut microbiomes. These enzymes are expressed in these subjects. But things got interesting when we started looking not for the known enzymes, but for some of these uncharacterized dehydroxylases. And specifically, we noticed that a group of uncharacterized dehydroxylases were actually the most prevalent and abundant members of the family in the actual human microbiome data sets. And so, again, here we're looking at prevalence. This group of sequences really stood out as being also found most widely among the different participants in this cohort. So this certainly stood out as a gene or protein of interest to try to characterize. But now, how do we actually connect this dehydroxylase of unknown function back to a specific catechol substrate and transformation? And so at this point, Min and Raj turned to the dietary information that we had on these subjects and did a bunch of different analyses and ended up learning that there was a strong correlation between having this dehydroxylase and consuming both coffee and cruciferous vegetables. This helped us narrow down the specific types of substrates that we might consider as candidates for this dehydroxylase to different types of polyphenols that are prevalent in those foods. And we used that protein structure prediction strategy with the uncharacterized enzyme to predict the size and shape of the substrate funnel. This helped us narrow our focus on hydrocaffeic acid and hydrocaffeic acid derivatives because it looked like this enzyme might have a smaller substrate. So, by combining all of these different approaches, what we were ultimately able to demonstrate from a biochemical perspective was that this unknown catechol dehydroxylase is actually metabolizing hydrocaffeic acid, which is an abundant catechol in these types of foods, is also a metabolite derived from metabolism of many more complex polyphenols. This enzyme is present in both cultured Gordonibacter strains and many uncultured gut bacteria as well. So moving forward, we're very interested to kind of understand the biological consequence of this transformation because of its high prevalence within human gut microbiomes. Min has an intriguing hypothesis that he's excited to explore. It turns out that hydrocaffeic acid itself has anti-inflammatory activity in animal models and in human tissue culture. But if you remove the hydroxyl group, this anti-inflammatory activity is greatly attenuated. And so now we're exploring the hypothesis in animal models that this dehydroxylation by the gut microbiome might actually be reducing the benefit of consuming this catechol. And we have some very preliminary analyses of the MLVS cohort that are consistent with this hypothesis, namely that if you look at levels of the inflammatory marker CRP in the cohort, and you also look at the dietary information from the cohort, you see that subjects who consume cruciferous vegetables but have a high level of Gordonibacter that can metabolize hydrocaffeic acid actually have higher inflammatory markers, levels of this inflammatory marker, than subjects consuming the same amount of vegetables that lack this activity. So much more work to do to explore this connection in the future. This work is going to be greatly enabled by some new tools that we've developed in the lab. So a really talented grad student, Young, has, after much effort, developed the first set of genetic tools for this family of gut actinobacteria. So we'll now be able to knock out this gene in isolates that we use in animal experiments. We also can now express these enzymes in native hosts to enable more biochemical and structural studies. So very excited about where this is headed. So I hope this first part of the presentation, highlighted some basic enzyme discovery, kind of give you a taste of some of the strategies we use to connect metabolism to enzymes, but also highlights how enzyme discovery can raise interesting questions of a biological nature to explore in the future. But the other question that, of course, this raises that will now connect to the second part of my talk is the question of we now understand this activity and how it happens, but how do we actually control it? Ideally, that's what we'd like to do if we'd like to establish causal links to the gut microbial metabolism and host phenotypes. And certainly from a therapeutic standpoint, whether it's promoting a beneficial activity or preventing a potentially harmful one, we need to think about strategies to control metabolism in complex communities. And so for the rest of the talk, I want to focus on a story that has evolved to that stage, where we're really thinking about studying biological activity of a microbial metabolite, but also developing strategies to control its production in complex settings. And so this connects back also to the project that I've worked on with Sylvia's group here at Minnesota. And this is the story of colibactin, a gut microbial metabolite that is connected to colorectal cancer. So just to briefly kind of summarize what we know about colibactin, this metabolite was first discovered in 2006 when the Oswald lab in France connected a set of uncharacterized genes in gut E. coli and other gut Enterobacteriaceae to genotoxic phenotypes. So co-culturing these bacteria with human cells resulted in genotoxic effects. This gene cluster and the organisms that carry it were then, in many subsequent studies, correlated with colorectal cancer. So PKS positive gut bacteria are found more frequently in colorectal cancer patients. And then various work in animal models has shown that this genomic region plays a causal role in tumor invasiveness and multiplicity. So of course, the question right from the beginning that everyone was interested in was, what are these genes doing? What are their functions? How are they leading to genotoxicity? So it was very clear at that point that the gene cluster was encoding a set of biosynthetic enzymes, specifically something called an enzymatic assembly line. Just like we use assembly lines to build cars and other complex machines, enzymatic assembly lines like non-ribosomal peptide synthetases and polypeptide synthases use the coordinated action of multiple enzymatic domains to build complex molecules. And of course, these types of assembly lines are well studied because they produce some of our most medicinally important natural products, most notably antibiotics. And so it was very reasonable to propose that the enzymatic assembly line encoded by the PKS genes was making some sort of complex small molecule genotoxin, and that presumed molecule was given a name, colibactin. So what would normally happen at this point was that a chemist would ideally use colibactin's genotoxic activity to guide isolation and structural characterization of the natural product before trying to study its bioactivity or biosynthesis. But those traditional types of approaches have never worked for colibactin. You'll see later this is likely due to the instability of this natural product. But my group and I were still very interested in this molecule, wanting to understand its structure and activity despite these obstacles. So we decided to take a very unconventional approach. We wondered whether we could study colibactin biosynthesis prior to isolation. We wondered if we could use our knowledge of other enzymatic assembly lines, bioinformatics, and our chemical intuition to predict the reactions that these enzymes would catalyze. We could imagine then testing our hypotheses by characterizing the enzymes in vitro and perhaps gaining some structural information. And we were able to use this reverse approach to obtain the first structural information about colibactin, and this prompted a number of other groups around the world to also join us in this effort to characterize the molecule using this unconventional approach. So many years of hard work by a really creative team of graduate students and postdocs, including Carolyn, Lee, Matt, and Yindi, ultimately provided us with enough information to predict a structure of colibactin. So here I just share that structure prediction. Again, we formulated this structure using knowledge from studies of biosynthetic enzymes, also isolation of fragments of colibactin from various mutants missing different PKS enzymes. So this structure is, as you'll see, a pseudo-symmetric structure. It's quite complicated. The key structural feature I want to highlight, after I point out that others have proposed a similar structure, are these two cyclopropane rings. So basically, these strained three-membered rings, really gave us a critical clue about how colibactin might be causing genotoxicity. Specifically, they suggested that perhaps colibactin was reacting directly with and damaging DNA. And so this is where we teamed up with Sylvia and Pete here at Minnesota. We wanted to characterize products of colibactin-mediated DNA damage, and we were able to successfully apply untargeted DNA adductomics to find colibactin-derived DNA adducts. And we were able to do subsequent experiments to show that these types of DNA adducts come from decomposition of larger lesions, specifically an interstrand crosslink. And that's consistent with our proposed colibactin structure, which has two cyclopropane rings. This molecular information about colibactin-mediated DNA damage has also been helpful in other contexts. Specifically, over the last few years, multiple groups have reported the discovery of mutational signatures that arise from colibactin exposure. These mutations are consistent with an interstrand crosslink, and very recently, again, with critical help from Sylvia here, we've been able to actually confirm the structure of the colibactin interstrand crosslink and its specificity for alkylation at these AT-rich regions. So the mutational signature work, I think, has fueled even more interest in colibactin and its role in cancer development, because now we can start to do things like look at cancer genomes and see evidence of colibactin exposure. And interestingly, we see mutational signatures not just in colorectal cancer genomes, but in other tumor types. One of the most interesting, I'll just point out, is oral squamous cell carcinomas. Head and neck cancers turn out to be the tumor types, at least in initial studies, where these mutational signatures are found most frequently. E. coli had been associated with these tumors prior to the discovery of mutational signatures independently. And I think most strikingly, these types of tumors occur very frequently in patients with Fanconi anemia who have deficiencies in interstrand crosslink repair. So collectively, the mutational signature work, I think, has really confirmed that humans are exposed to colibactin and has raised interesting new questions about in what contexts this exposure may be relevant for cancer development. So there's obviously much more work to do to understand many biological questions that have arisen from an understanding of colibactin structure and biological activity. But the one I want to talk about just briefly to end the seminar is our work thinking about opportunities for therapeutic intervention. And so this is the work of a former graduate student in the lab, Matt Volpe, and we were able to collaborate with a really terrific structural collaborator, my colleague at Harvard, Rochelle Godet, and her former grad student, Jose Vaila. And in thinking about how to prevent colibactin production, we became very interested in the idea of interfering with colibactin biosynthesis. So I didn't take you through the whole biosynthetic pathway. I had just shown you the final structure of the molecule that we predict. But I want to now share a critical final step in colibactin biosynthesis that has provided a great inroads into manipulating this pathway. So this is the final step in colibactin biosynthesis, and it's catalyzed by an enzyme that was actually one of the first in the pathway that we studied. So this enzyme CLBP on the surface is a pretty simple enzyme. It's a serine peptidase, and it's located in the periplasm of bacterial cells. And that localization, I think, is very important. What CLBP does, and we showed this biochemically, is to very specifically catalyze amide bond hydrolysis. But it does so just at these specific N-myristoyl D-asparagine structural motifs. So it removes these two parts of a inactive precursor molecule we've named precolibactin. And this then triggers a beautiful cascade of cyclization reactions that end up generating those reactive cyclopropane warheads that I showed earlier. And so we think that this biosynthetic step probably evolved as a way for the producing organism to protect itself from colibactin. So the organism makes an inactive molecule, exports it out of the cell away from its own DNA, and then generates these reactive groups. So Matt became very excited about the idea of trying to interfere with colibactin production by inhibiting the activity of this serine peptidase. I think this was a good choice for a couple of reasons. The enzyme's periplasmic location might make it more accessible to small molecule inhibitors. And being a serine peptidase, we could maybe leverage a lot of known strategies for developing these types of molecules. So Matt started initially by just surveying the enzyme's activity towards a bunch of different substrates to better understand its activity. And we found that while you can make big changes to the acyl tail of the molecule and make lots of changes to the C terminus of this type of substrate, you had to have the D-asparagine side chain, both the functional group and the stereochemistry intact adjacent to this cis-acyl amide bond in order for the enzyme to function. So what Matt did initially to try to find inhibitors was to then start assaying the enzyme in the presence of molecules that were known to be known to inhibit related enzymes. So other members of the broader family, or molecules that were just known to be broad spectrum serine hydrolase inhibitors. And what was kind of surprising was that none of these inhibitors, even the very broad spectrum ones, showed any activity towards CLBP. The other thing I'll just mention is that these two compounds, which were reported to be CLBP inhibitors, but had never actually been assayed against the purified enzyme, were totally inactive. So we couldn't find a starting place based on this survey of existing inhibitors. And so what Matt had to do instead was to think about this critical sort of prodrug motif, this N-acylated D-asparagine that was so critical for enzyme activity, and design an inhibitor based on that structural motif. And so the design strategy was pretty simple. He decided to try to install a boronic acid into this structural motif with the idea that we might be able to trap that nucleophilic catalytic serine in a reversible covalent bond, impairing enzyme activity. And this turned out to work very well. All of the inhibitors we were able to synthesize have very good potency, both towards the purified enzyme and in bacterial cultures. So with Rochelle's help, we were able to actually characterize how our inhibitor interacts with the CLBP enzyme. So this is a crystal structure of the enzyme. You can see that not only is it periplasmic, but it's anchored in the inner membrane of bacterial cells. And these transmembrane helices actually are very important for the enzyme to function. Here's the active site at the interface of these two domains. And these transmembrane helices, we think, interact with that long myristoyl acyl tail on the substrate. So if we look more closely at the inhibitor in the enzyme active site, we see indeed there's a covalent linkage between the boron atom and that key catalytic serine supporting our proposed mode of action. If you rotate the structure to look a little bit at that interaction between the deasparagine side chain amide and the protein, you see a very exquisite network of hydrogen bonds we think account for the high specificity of the enzyme. So with this inhibitor, various inhibitors identified, we were then able to move forward and learn a little bit about the inhibitor mode of action and how it could be applied. Here, we're just performing some metabolite profiling experiments that demonstrated that treating colibactin-producing organisms with the inhibitor basically phenocopies genetically deleting CLBP, which supports the idea that this inhibitor is quite selective. It's not interfering with other types of metabolic pathways. We have applied the inhibitor ex vivo to more complex samples. Here we're taking a mouse fecal sample that lacks Enterobacteriaceae, adding in our colibactin-producing E. coli, and discovering that indeed the inhibitor works very well to prevent that prodrug activation step in this very complex setting. And finally, and most importantly, we've also shown that our inhibitors prevent the genotoxic effects of colibactin-producing organisms in human cell culture. So inhibitor treatment prevents G2M phase cell cycle arrest, which is a hallmark of colibactin exposure. It also prevents the activation of interstrand crosslink repair as assessed by examining ubiquitinylation of the Fanconi anemia pathway protein FANCD2. And I'll just note the specificity of this effect for colibactin. So you can see that here inhibitor treatment has no effect when we expose cells to other cross-linking agents like mitomycin C or cisplatin, consistent with the hypothesis that we're really interfering with colibactin production specifically. Then finally, with help from Sylvia's lab, we were able to measure DNA adducts in the presence and absence of the inhibitor and find that indeed DNA damage is lessened. So now we're very excited to explore the utility of this inhibitor in the context of animal studies. Specifically, the ability to control when we dose the inhibitor might allow us to modulate the timing of exposure to colibactin and elucidate whether there are critical windows where this genotoxic insult is important for tumor development. We also think perhaps this could inform some strategies for cancer prevention or interception, particularly for patients like Fanconi anemia patients who may be at much higher risk for cancer development. Okay, so I hope that this presentation has shown how linking microbial metabolism to enzymes can be really critical in the context of microbiome studies. Not only does it allow you to better and more accurately identify the functional capabilities of microbiomes from sequencing data, but it provides inroads to manipulating microbial functions in complex habitats, whether it's through the development of inhibitors of those enzymes or by helping you to select or engineer probiotic strains that have specific metabolic capabilities. Moving forward, we're very excited to continue to discover enzymes that are involved in important biological processes. We're very interested in further applying inhibitors both to study microbial metabolism and to help to perhaps even discover new metabolites that are made using particular bioaesthetic enzymes. And I'll also point out, in case anyone is interested, that my lab is moving into other areas of the human microbiome as well, most notably the vaginal microbiome, which has very strong links to women's health. And so with that, I'll end. I'll just thank the whole lab. I've acknowledged the individual lab members who worked on projects as I went along, but I'm really grateful to the whole lab for really being such an amazing team of colleagues to work with. I'd like to acknowledge all of our collaborators, particularly our collaborators here at Minnesota on the colibactin project. Thank our funding sources, and I would be happy to answer any questions.
So I have a microphone for anyone who has a question.
In your presentation, you showed how the specificity of the catechol dehydroxylase really came from that cone structure. For the enzymes with larger substrates, do you think it would show the same level of specificity as that kind of cone region expands to accommodate the substrate?
Yeah, so that's really interesting. So I would say we see a correlation between size of the cone and the size of the substrate. What we haven't yet elucidated, and I think it will take crystal structures to really, or cryo-EM structures to really pin this down, is how are the actual interactions with specific parts of the substrate mediating binding to the molybdenum. I think that's what's going to control selectivity. You could imagine that with a big funnel, you may be able to fit smaller molecules in there, but are they going to be able to bind with the precise orientation that's needed to access the molybdenum? I think that's where most likely we're not going to see a broader, broader substrate tolerance. And preliminary assays we've done with things like the catechin dehydroxylase support that hypothesis that a bigger active site doesn't correspond to any gain in breadth of substrates.
Thank you.
So humans are hindgut fermenters. So most of our microbes are at the very end of the digestive tract. And a lot of digestion and absorption happens way before that.
Yeah.
So in your whatever diet study, do we have any serum or circulating metabolites to show that these microbially derived metabolites correlate with the input of substrate?
Yeah, so for the MLVS cohort specifically, we don't. There are some studies that have matched serum metabolome profiles. None that we've been able to find that also have metatranscriptomic data sets associated with them. So for these specific dehydroxylated products, what I can say is that in serum metabolomes, you can detect these things. What we don't yet have from the perspective of our work is a good cohort to really kind of correlate changes in serum or urine metabolite profiles with individual microbial enzymes. What I will say about localization, just because that's very interesting, where is this metabolism taking place? Is it really in the colon or is it in the small intestine? So for Agarthella species in particular, there's some indirect evidence that they may be present in the small intestine. They're involved in the metabolism of drugs like digoxin that are absorbed in the small intestine. So while there's limited direct evidence for their biogeography, I think it's certainly a possibility that they may be there in low abundance at the actual site where a lot of these compounds are being absorbed.
Go ahead.
Thank you. Very excellent talk. I had a question about colobactin. You touched on how because the prodrug is sort of metabolized in the periplasm, that this is a sort of a defense mechanism for the E. coli, but the E. coli is not by itself, it's in a microbiome. So do other bacteria in the microbiome have some sort of defense mechanism against colobactin?
Yes, so that is a great question. I don't have a slide in this slide deck. One of the things we are very interested in exploring is so the effects of colibactin on other members of the microbiome. So colibactin producers themselves have a backup resistance mechanism. So if the toxin makes it back into E. coli, there is a protein that is, I would say it's incompletely characterized. There's some evidence it may hydrolyze the cyclopropanes. It also binds to DNA and maybe sort of possibly having a repair function. What it does is still unclear. But this protein CLBS is found in other organisms that can't make colibactin. And we've recently done some studies to show that these CLBS homologs in other organisms are functional. They can prevent DNA damage to colibactin and other bacteria. And that's something actually also in our study we showed that, to me, at least a surprisingly large range of bacteria are susceptible to colibactin. That's consistent, I think, with what the field is kind of learning, is that colibactin production probably evolved as a way for these organisms to target other microbes, not primarily to target the human host.
Go ahead and take the question online.
Okay, so one of these questions from Tian, I answered, why do bacteria produce colibactin? So another question from Rick, what are your thoughts about how enzymatic activity might change under different environments of different tissue environments? Will enzyme function be different in different environments? I think that's a great question. There will be, I think, various environmental factors that will change the expression of enzymes or their activity. So for example, these catechol dehydroxylases I mentioned are air sensitive. And so if you have organisms living in very close proximity to the gut epithelial surface, or if you have an inflamed gut epithelium where you have higher levels of oxygen or reactive oxygen species, most likely those enzymes will be less functional. And yes, certainly the presence of specific substrates that might be, especially host-associated substrates that might be present at in different areas, different geographic regions of the gut might certainly influence what enzymatic activity is present.
All right, let's take our last question with Rick down front.
Yeah, oh, fantastic talk. On the calbactin structure, it's reminiscent of bleomycin to a certain extent in that central part.
Yeah.
What do you know? Do you have intercalation within the base pairs? And then if you just take how important is that placement within the DNA to the actual activation then of the cyclopropyl groups? Have you been able to show any DNA binding of just the piece, of just the side warhead, or do they need to be in that position in order actually for you to get cross-linking? So they're not actually that, they're not actually that active unless they're in that in that position.
Yeah, so this is very interesting. So my group has worked largely with the native colibactin when studying DNA alkylation, DNA recognition, binding, and specificity of alkylation. Seth Herzen and Jason Crawford at Yale have made synthetic analogs. And so if you piece together everybody's work, including, well, a bunch of our work on this subject is still in progress. What we're kind of learning is that the central part of the molecule is actually appear to be very important for sequence specificity. And so if you alter that part of the molecule, or if you break the molecule apart and have just one warhead, you don't see the same degree of sequence specificity or affinity. So when we've worked with the individual, like a half of colibactin, we see, just much less potency. You need to add high concentrations of those colibactin mimics to see actual DNA alkylation. Yeah, we have been working on actually characterizing a structure, not ready to be shared yet, but hopefully soon. Yeah, I think it is really what we're seeing is that the entire structure is very important for DNA binding and specificity.
Sequence specificity, can you share anything about that? Is there is it translated anything where, with regard to human?
Yeah, so you could kind of indirectly infer the sequence specificity from where the mutational signatures were. So this is actually work from a former Minnesota graduate student, Eric Carlson, who was a postdoc in my lab. And again, we're hopefully going to be working on writing this up very soon. But generally, what I can say is that experimentally, in our hands, we see the sequence specificity you would predict from mutational signatures. If anything, we see a little bit broader specificity. So we're seeing a cross-link forming between two adenines 4 base pairs apart. The sequence context is important, although we see a slightly broader tolerance for GC base pairs in and around that site. We think there may be repair processes at play in some sequence contexts that may not be as effective in other contexts. And as you would predict from the end site of alkylation, N3, colibactin's a minor groove binder. Yeah, it's I think those general things I think are things one would have probably predicted from mutational signatures, but it's nice to actually confirm them experimentally. Yeah, and Eric's done terrific work on this. So I hope to be able to share some of that pretty soon.
All right, well, thank you so much. One more round of appreciation for a great talk. Thank you.